All right, everyone. Thank you for joining us at tonight's After Hours, everyone in person and online. Welcome. We have Dr. Nicole Millette, Assistant Professor at VIMS. She is originally from Minnesota, but she's been moving up and down the East Coast for her education. In 2011, she received her Bachelor of Science in Marine Biology from the University of Rhode Island. And in 2016, she received her PhD from the University of Maryland Center for Environmental Science. And between 2016 and 2020, Dr. Millette completed 2 postdoc positions in Miami at a NOAA lab and at Woods Hole, Massachusetts at the Woods Hole Oceanographic Institute. Dr. Millette joined us in 2020, and her background is in phytoplankton ecology, understanding how phytoplankton blooms form, how changes in phytoplankton impact other parts of the food web, and using phytoplankton to better understand ecosystems. And during the pandemic, Dr. Millette, who knew no one, shared her love for Muppets and trivia with us by hosting virtual trivia for VIMSers. So she put it together. It was very creative and it was really great and inspiring. And we learned a lot of great Muppets trivia. So if you'll please welcome Dr. Millette.
All right, thank you, Candace, for that introduction. Get my laser pointer set up here. All right, hi, yes, I like to go by Nicole, so I'm Nicole here, and I'll be talking to you tonight about winter phytoplankton blooms and how they connect to potentially striped bass in the upper part of Chesapeake Bay. So what I want to start with is just asking everyone to think about what they picture when they hear the word winter. So as Candace mentioned, I'm from Minnesota. So for me, winter looks something like this, sub-zero temperatures, a lot of snow, dark, cold, short days, and maybe not this extreme in Virginia, but a lot of the same ideas I'm sure come to mind to people when they think about winter. So the day lengths are shorter, there's less sunlight, and then just the intensity of the sunlight is less. And then also, of course, temperatures do drop. And what you see around you is there's not leaves on the tree, the grass is dying, there's not new plants growing. There's definitely plenty of animals that migrate to the south of us or things that decide to go into hibernation. But that's all based on what we see here on the land. And there's really the assumption that what happens on the land is very similar to what's going on in the water. Things become too cold for anything to really grow, and it's too dark for any of the plant life to really grow. And honestly, who could blame anyone for thinking that when this is what the water can look like during winter or something like this? It doesn't look like there should be a lot going on there. And of course, there is truth to that idea. There's plenty of examples of organisms within Chesapeake Bay that move to other parts of the ocean. Your crabs and your lobsters will move into deeper waters farther offshore. There's types of phytoplankton that they form these resting stages. They just build a little case around themselves and they sink out and they settle into the sediment and they just take a little nap until they get better environmental conditions for them to keep growing. However, that's not entirely the whole story of what's going on in the winter time in the water. And in fact, there can be a surprising amount of biological activity during the winter time. Especially, there can be these winter phytoplankton blooms that form that a lot of people really don't seem to take notice of, partially because no one's going out sampling during these conditions either. But when people think about phytoplankton blooms, they usually associate that with warmer temperatures. For example, we have a very large phytoplankton bloom going on right now in the York River just outside. But there are in fact comparable blooms that happen in the winter time. And so the obvious question that comes from that is, if there's these winter phytoplankton blooms and they're growing, which means they're photosynthesizing, which means they're producing new carbon into the water, what's happening to all of that new carbon that's coming into the water? Is there other biological life that's benefiting from it? Or is there really no other life that's going on in the winter time and it just kind of all goes to waste? So that's what I will be talking about today. Looking at these winter blooms, how these winter blooms might benefit other organisms in the water, and ultimately how winter blooms might be impacting other parts or other times of year and other parts of the food web. So I'd like to introduce you to your cast for this evening of who I will be talking about. First of course, we have our winter bloomer themselves, Heterocapsa rotundata, this little cutie right here. So I will be using the term Heterocapsa a lot. That will be referring to the winter bloom. A species right at the cusp of everything here, right at the center, is called Eurytemora carolleeae. This is a type of copepod that is very central to eating phytoplankton like Heterocapsa, and then themselves getting eaten by different life stages of fish. So they're a very important connection between phytoplankton and other parts of the food web. Now, Eurytemora is a crustacean, which means it molts. It has multiple life stages and it has to shed its exoskeleton to go from one life stage to the next. And this particular species has 12 distinct life stages, 6 of which look like this little alien up here, kind of a water fleet type thing. It's called a nauplii. Well, the last 6 stages are called a copepodite stage, and they look more akin to this. I show both of these pictures because I will be talking about both of the different life stages, and I will make the distinction as clear as possible when I'm referring to what the nauplii life stages are doing compared to the copepodite life stages. And then last of all, of course, I will be talking about striped bass, considering that's what we're all building up to. So to get started with our winter bloom species, Heterocapsa rotundata. I studied the species for my dissertation, and I happen to have 3 research papers out on it. And it's not a species that gets a lot of attention from the larger community. So because of that, congratulations, you're speaking to the World Heterocapsa expert tonight. Yay, how special. So feel free to ask me anything about it. So what I want to emphasize right away is that where these blooms of Heterocapsa form. So this is just looking at their average abundances within Chesapeake Bay across winter, spring, summer, and fall at different stations within Chesapeake Bay. Very clearly, you can see, first of all, they are very abundant in the winter time, but it's also this upper part of Chesapeake Bay, this Maryland region, which is why I'm focusing on the upper part of Chesapeake Bay. We won't be talking too much about this Virginia region because this particular species doesn't seem to have the same ability to form blooms down here, but that's a whole other story for another day. So what I want to help people understand is exactly how these blooms form, and really emphasize the conditions it takes for these blooms to form. But before we get into that, let's just, at a very basic level, how does any bloom form? And it's really, at its core, the idea of how any population grows. So if you have a population of anything, including humans, you have some amount being added to the population, some amount being taken away from the population. And if you have more being taken away than that's being added, your population's dying. But if you have more that's being added than being taken away, your population is growing. And if you're a phytoplankton, eventually you will find yourself with a bloom. Now, when talking about phytoplankton, we usually refer to them being added in terms of their growth rates here. How much is being added through growth? And we're referring to how much is being removed through grazing from predators. So if you have a bloom that's forming because you have your growth and your grazing are in balance. And then the growth rate is the factor that increases in order to start blooming. That means something in the environment has likely changed. They have more nutrients, they have more light, they have better temperature, something, but it's a bottom up control. Something from the environment beneath it is coming up to make better conditions for it to grow. Alternatively, if what's causing the bloom to form is the grazing is decreasing, that's considered a top-down control. Something from above them in the food web is changing and coming down so that there is less grazing, and now a bloom can also form in there because growth is now higher than grazing. I'll be talking about how both of these factors play a role in Heterocapsa forming their blooms. So first of all, let's tackle this messy figure over here. I didn't come up with this figure, thankfully, because it's a little bit of a nightmare to look at. But this was research done on Heterocapsa actually over 30 years ago in the Potomac River. And what they did was they went out, collected samples under different conditions. And you have here the temperature that that sample was collected at, the salinity that that sample was collected at, and this very long name here, which boils down to how stable the water column was when the sample was collected. So if the sample was collected over here at a value of 1, that means that the temperature and the salinity at the surface of the water were completely the same as they were at the bottom of the water. And basically the water column was not stable at all, and it was mixing around entirely from top to bottom. And then as this value decreases, there becomes an increasing difference between the temperature and salinity at the surface compared to the bottom, and it becomes more and more stable, leading to what some people might be familiar with the term stratification in the water. And what this paper found was that there was 2 groups that the samples fell into. This group over here, you have higher salinity and you have almost no stability in the water column, mixing around a lot. Well, over here, this grouping, you have lower salinity and this water column is a bit more stable. There's not so much mixing going on. It's a calmer water column. And what defines these 2 groups in terms of what was in these samples was these samples with the lower salinity. This is where Heterocapsa was dominating the population and a bloom was forming. So what this was telling us was when you have low salinity in the water, what's causing that is rainfall and then freshwater runoff. That's where you get lower salinity. That means there's less salt that's coming from a freshwater input somewhere. And usually when you have more river flow bringing fresh water or just straight off from the land, more fresh water coming in, that's associated with more nutrients coming into the system. And phytoplankton love nutrients coming into the system. This is your nitrogen and your phosphorus, and they need that to grow. So this can help them grow, a bottom-up effect. What I looked at myself, because this research had already been done, was this idea then of top-down controls, grazing on these blooms. So I set up these experiments once a week to look at how much of this Heterocapsa population was being consumed by different grazers. And this was data that was collected in 2013 in the Chesapeake River over on the eastern shore of Maryland. And again, a lot going on in this figure, but what we need to focus on is this line graph refers to the abundance of Heterocapsa. So a higher number here, more Heterocapsa in the water. This graph, the line graph, refers to the percentage of this Heterocapsa abundance that was being removed that week through grazers. And I was looking at 2 different grazers. This black line is our friend Eurytemora, because they are present and a very important grazer. And there was also another group called zooplankton that I won't be getting into, but they're just small zooplankton. And what you can see from this figure, first of all, this year there was not a bloom of Heterocapsa. There was a peak right here in abundance, but these are not high abundances. There was not a bloom. But what you can see was there was a lot of grazing going on. A high percentage of the standing stock every day, or the abundance every day, was being removed. Earlier on in this winter, there was somewhere between 80 to over 100% being removed. And then even later on, there was still between 40 to 60% being removed. That's a lot. Alternatively, for comparison, in 2014, now there was a bloom that formed. You can see these abundance numbers here. We got 20,000 compared to 4,000 here. Things got much more abundant. But starting here at week 5, you start to see consistently exponential growth or increase in the abundances of Heterocapsa. And the other thing that really stands out is that there's a lot less grazing going on. There was definitely about, that 40 to 60% earlier on in the winter, but starting around week 5 here, at the end of January, basically there's not a lot of grazing that's happening. It kind of just goes away. And the reason potentially for that I hypothesized was related to temperature. So you can see here, this is the temperature that was measured when the water was collected. And here is week 4 results. We still had some grazing, but then week 5 comes along and this temperature drops down to 0 degrees Celsius, or known as freezing. So 32 degrees Fahrenheit. And it stays very close to 0 for 4 weeks. And it's these 4 weeks right here in which the bloom really takes off and forms. So temperatures got so low that the grazers couldn't keep grazing. It was too cold for them to keep feeding, essentially. So that formed an opportunity for this Heterocapsa. It was still growing, so it made a bloom because there wasn't anything eating it. So that's kind of that's basically how these blooms form. It's really important to remember as I move forward that they need rainfall. So you need to have a winter where there's rainfall that's happening, precipitation, so that you've got new nutrients coming into the system for the Heterocapsa to grow. But you also need a reduction in grazing that's likely coming from low temperatures. So a year where you have a very cold winter with a good amount of rainfall, probably the right conditions for these blooms to form. That'll be important to keep in mind as I get later on in the talk, but trust me, I will be revisiting that. So great, we have a bloom that's formed. We did it. We have all of this new carbon in the water and it's got to go somewhere. And the obvious place is, I mentioned earlier, this copepod, Eurytemora carolleeae, very, very important grazer in the water column. As I said, they eat the phytoplankton, and then other things love to eat these copepods. So it is a key conduit of getting things from the phytoplankton throughout the rest of the food web. So the next logical question is, how does the bloom benefit this specific species that is present when the bloom forms? And one of the first things that became obvious to me was that the bloom's not impacting those later stages, those copepodite stages that look like this. I was going out and I was just tracking what's the average abundance every day. Every time I was collecting samples, what's their average abundance throughout the winter. And for every winter that I was collecting that data, there wasn't a lot of variability. It was very consistent, somewhere between 8 to 10 copepods per liter were present. That's just what was out there. There was an exception here in 2012, which I will talk about at the end of the talk. But it didn't look like this bloom was affecting the later stages for a variety of reasons. But there's still the question of how do they impact these earlier stages, these nauplii stages, because these stages, they're very vulnerable to starvation. They're young, they don't have a lot of fat on them that they can use if there's not food available. So they need food around, and they're to keep living. So that's what I looked at, was so this was all stuff now done in the laboratory. If I took a culture that had these little nauplii in it, and I fed them different concentrations of Heterocapsa, how did that affect their mortality, the rate at which they died? And first of all, unsurprisingly, it turns out over a 2-week period, if you don't give the nauplii any food, pretty much all of them die. So good place to start. Second, if you give them just a little bit of food in the form of Heterocapsa, their mortality greatly decreases. So that means that Heterocapsa is not detrimental to them in any way. It's a good food source. But it continues to decline as you give them more and more food. And somewhere around this, 2,000, maybe 5,000 cells per milliliter, you really, really just get rid of most of the mortality that would be due to starvation. There's plenty of natural mortality that's going to happen. You just can't account for it. But looks like having more Heterocapsa around is going to mean more of these nauplii in the water are surviving. That's a good thing. So just to reiterate where we've gotten so far, you get a winter, it's cold, it's got rainfall, you got yourself a bloom, and now that bloom's providing food to these young stages of Eurytemora, the nauplii stages. So more of them are surviving to later stages. And now, finally, after all of this buildup, is when we get to where the striped bass finally come into the picture. So striped bass, also known as rockfish, have to mention that. I'm not going to be talking about, these big trophy sizes or even these, cute little juvenile versions. I'm going to be talking about the alien versions of striped bass down here. These are the larval stages. This is what they look like right after they have hatched from the eggs, and they have just used up the last of their yolk sac. They need to feed at this point. And if they don't have food available, they're pretty quickly going to die. And that is who I am talking about. So if people aren't aware, striped bass actually have a very wide geographical range all over the East Coast, down into the Gulf of Mexico, and they even have some introduced range over here on the West Coast. But one of where they're most well known is here in Chesapeake Bay. Now, their life history is that they come up and they spawn in freshwater environments during the springtime. So that's what they use Chesapeake Bay for. Otherwise, you're going to find them in these more offshore areas. And Chesapeake Bay has a lot of great places for them to spawn because we have so many of these rivers, tributaries that feed into Chesapeake Bay itself, plenty of freshwater places. So it's a great place for them to spawn. And then the young that have spawned or that have hatched that year will then grow up within Chesapeake Bay until they're large enough, and then they will also leave into the offshore open ocean region. So I'll be using this word recruitment up here a lot. So I wanted to talk a little bit about that. Once a fish larvae reaches a certain size, it's considered large enough that its mortality rate is so low that you can consider it recruited to the adult population. Its chances of joining the adult population is almost a foregone conclusion at that point. And so this is when the newly hatched larvae of any year get large enough that they are going to be recruited to the adult population. And believe it or not, people are very interested in understanding what causes variability in this recruitment each year. So in Maryland specifically, that's what we're talking about. The Maryland DNR goes out and they does these surveys between July and October. They're just taking these seine nets and they're collecting and counting how many young new striped bass there are for that year. And they're taking the average of those measurements that they're taking, and those are called, they make a calculation referred to as a recruitment index. And that's what this figure here is showing. This is for the average mean of these new striped bass later in the year after they've spawned for all the different locations that the Maryland DNR does their surveys. And there's a lot of variability within each year. So depending upon the spawning location, some areas can have more successful recruitment compared to others. And then also between years, you can get a lot of variability. So people, of course, want to know what's causing the variability in this recruitment. How do you get a lot of the larvae to be recruited to the adult population? Because then you're going to have more adults, and that's what people want. And there's the obvious idea is that if there is more food available to these larvae when they start feeding, that more of them are going to survive. It's a very intuitive idea, but scientists had to put a name to it anyway, and we call it the match-mismatch hypothesis. So if fish larvae match up with their prey in time and space, then they are going to have better survival than if they mismatch with their prey in time and space. Excuse me. But it gets a lot more complicated than that very quickly. How do they match up with their prey in time and space? They don't have that much control over it. But one thing that people have found is that down here, this is showing when zooplankton prey have their peak abundance every year. So this is showing just day of the year. So this is April 30th over here. How does that relate to what how successful recruitment was for that year? And essentially, if the peak zooplankton abundance occurs later in spring, for some reason, recruitment is better. So now we have a new question to ask. It's not what makes what's good for recruitment. It's how do you get abundances of the prey to happen later in the spring? And of course, the key prey item here is our friend Eurytemora. That's why I've been talking about it. So the striped bass larvae love to eat the copepodite forms of Eurytemora. So we want a lot of those apparently later in the springtime. And Eurytemora usually forms peaks of abundance in spring. That's been shown before by other people. So just for example here, this is the peak in April that forms of Eurytemora up here. But it's not, I mean, the timing of when this occurs in April, or it can also occur in March, May, there's a lot of variability there. That's what we're trying to understand. So when does this peak exactly occur? And for me, my advisor in this case came to me and said, what about water temperature? So what about temperature? Why would he suggest this? Well, first of all, he was author on a paper that talked about temperature. So he thought it was a great idea. But copepods the rates, just their biological rates, are very closely tied to temperature, including the rate at which they develop and go through each of their different stages. So if it's warmer, they're going to be molting much more frequently and going through each of their stages. If it's colder, they're not going to be molting as frequently. More importantly, if you have like really cold temperatures, you've got very slow developmental rates. But if you increase that temperature by a degree or so, you're going to drastically change their developmental rates, because this is an exponential relationship. So it takes a lot to change that rate at these higher temperatures, but at these low temperatures, things are changing very quickly in response to temperature. So what we have, first of all, is a lot of variability between years in the average temperature. You can have things near an average temperature very close to freezing. Meanwhile, you can have average temperatures that are downright balmy in comparison. So lots of variability in your winter temperature. And copepod developmental rates should be responding quite drastically to that. So what happens if these nauplii versions of the Eurytemora that hatch in wintertime and develop through wintertime, how does variability in the temperature that they're developing under in winter impact the time that peak abundances form of the later stages, these copepodite stages, come springtime? Is there an influence on there? And that's what I was looking at. So I formed this is a visual representation of a very simple model I created to look at that. And what I put into this model was a daily temperature rate. So I started a model run. And on that very first day, it would feed in for whatever year I gave the data. What was that temperature on January 1st? And it would immediately then calculate what's the daily fractional development that nauplii that hatched that day. How much did it develop of the proportion of the developments that it has to go through? And I calculated that every day. And then what the model itself was doing was adding in every day what I called a cohort of nauplii. Basically, I was saying every day there has to be at least one nauplii that hatched out in the environment. I don't care how many, but for a nauplii hatched on any specific day, how long would it take it to go through all of its developmental stages and reach the adult stage? So I would be following what fraction of its development it would be going through based on the temperature until it reached 1. So I added in a total of 78 cohorts up until March 19th in the model run. And I ended it there because that's the last day of winter. So after that springtime, and I just kept the model running until all of these cohorts reached the adult stage. I also, my calculator. I'm not going to talk too much about this, but another thing I did was go into create a very, very crude estimation of relative abundance and then added mortality on there. The reason I don't want to talk about this too much is because these aren't meant to be real abundances. What I'm meant to show is later on a comparison of how the timing of when these peaks occur based on the developmental rates throughout winter might also impact the abundance of the peak that forms, but they are not representative of what a real abundance of that peak would be, merely a comparison to other results that models put out. So I don't want people to focus on the abundance aspect. So for the actual model runs that I did, I had to get daily temperature data, which I wasn't collecting, at least not enough of it. But there's NOAA stations that are located all throughout Chesapeake Bay, and they're collecting data every 6 minutes. So I was able to download that data and average it to a daily temperature rate. And I got that data from a location in the Choptank River, the Patuxent River, and the upper parts of Chesapeake Bay near Tolchester Beach. All of those are located with systems with known spawning grounds of the striped bass. So I was able to, this is the number of years I was able to get enough data to do these model runs for. It's not clearly gaps in there because there's times when the instruments malfunction and there's not enough data to actually do a model run. But this is enough data. I also took from USGS discharge flow rates because we're talking about winter temperature. And so it only makes sense at some point to put in flow rates out of these rivers that are related to rainfall, because that's also important to something I was saying earlier. So it seemed necessary to tie back. So for these, this data, this is just looking at what the daily water temperature was in these 3 different systems. And then this dark line here is the average of all of this data. So you can see there's, again, a lot of variability in the temperature between years and then within years. But there also is a difference between the systems. It's not easy to pick out right away, but you can look at the starting temperatures on January 1st for each of these systems, and the average temperature for the upper Chesapeake Bay is well below 5 degrees Celsius. Well, for the Choptank River, it's around 5 degrees, and it's actually closer to 7 degrees for the Patuxent. So these are ordered in terms of increasing average temperature. But I did a lot of these model runs, and there's no reason to show you all of that data. It would just be a whole mess of information up there. So I focused on the results from that running average here of all of the temperatures for each location, as well as the results for the warmest average year and the coldest average year at each location. And this here is the daily fractional developmental rate that the model run was calculating every year. And again, it's just showing you how much variability there is between years and within years. So this data is directly related to temperature because temperature is what was being used to calculate it. And these are our results here. So this is, again, down here, the day of the year, just any day of the year from starting in January 1st to April 30th for a model run. And then this is that relative abundance. That's not a real abundance, but if it's just higher here, there could theoretically be more of these copepods. But starting, every model over here starts with the nuclei that are being hatched. And as it goes along, as soon as you start adding to this relative abundance, that's when the adult versions, the very last stage of Eurytemora, is showing up. So that's all it's being shown here in these figures. And these results are for the warmest here in these first ones, the average, and then the coldest. And there's a couple things that should jump out. First of all, the first cohort to reach the adult stage in these warmer model runs can happen about 40 days before when a cohort reaches the adult stage during these colder years. Over a month difference of how long it takes these groups to develop. It's very much slowed down. But also these cold run, the results here from the coldest years, you get a really high peak in this relative abundance. Essentially, all of these nuclei that were hatched, they were developing so slowly that they just kind of all coalesced around the same developmental stages. And very late in the year, as the temperatures increased, all of a sudden they all started to reach the same developmental stage at the same time. So they hatched over a 78-day period, but they all reached the adult stage within a few weeks at the very end of spring. So what you've got here is what's hatching in the winter time is going on to becoming food for these striped bass. And the last part of new data here that I want to go into is what I looked at was I compared the recruitment index results that were collected by the Maryland DNR, and I looked for relationships with model output data that I had. So how did they relate to how temperature was influencing the copepods in the model? And I also added in, were they at all related to the discharge rates? So in summary, what is being shown is that at each of these locations, if you have higher average discharge rates in between January and March, along with various aspects of how the copepods are impacted by colder winter temperatures, that's a good thing for striped bass recruitment. So striped bass recruitment also wants higher amounts of rainfall or discharge coming in Chesapeake Bay during the winter time. And it also wants colder temperatures because that's going to benefit at least the colder temperatures are going to benefit their prey. So in summary, for the striped bass aspect, you have these cold, if you have a year with cold temperatures, there's a really high probability that you're going to have good striped bass recruitment. And I'm specifically referring to the winter temperatures here, not necessarily the spring temperatures when they actually spawn. If you have a year where there's higher winter temperatures and there's not a lot of rainfall, very, very high chance that there's going to be poor recruitment that year. It's not going to be good for the striped bass larvae survival. If you have a warm year, but there's rainfall associated with that warm winter, there's probably going to be a decent amount of recruitment. It's not going to be abysmal. There will be some recruitment that happens, but it's not going to be great. But why? Why does this happen? Why is all of this happening for the striped bass? I'm talking about winter time and striped bass spawning in the spring. Let's tie everything together in a nice, neat little bow. So first of all, these warm, dry winters. Going back to this figure here, way back at the beginning, I told you that copepods, these Eurytemora abundances, for the most part, didn't change during the winter time, with the exception of 1 year here. Well, this was a warm, dry year. Temperature here was really hot. And I can tell you from experience, it was a very dry year. Could not get rainfall for the whole year, basically. But it really, really hurt the phytoplankton abundance. So there was barely any phytoplankton in the water. It was so low that it was actually affecting the copepodite stages of Eurytemora. These little guys here. So these warm, dry years, phytoplankton concentrations in winter are so low that you don't have a lot of adult or later stage Eurytemora. So they're not going to be producing a lot of these nauplii for hatching. And even when these nauplii do hatch, they don't have a lot of food to feed upon. So they're going to be dying. And then as they go throughout winter developing, the few that are still developing, they're going to be reaching the adult stage earlier in the springtime and over a longer period of time. So just a couple new ones will be showing up in the population. So when these striped bass larvae here need to start feeding, they're not going to have a lot of food available to them. And the recruitment to the adult stage is going to be really, really bad for these warm, dry years. You have these warm, wet years, you have more phytoplankton because you have rainfall happening. So there's more nutrients in the system. So more for the phytoplankton. So they're a little bit happier, but you're still not getting a bloom that forms. But the copepodites here, they are able to hatch more nauplii, and more of the nauplii will be surviving. They have a decent amount of food, but again, because the temperatures are warmer, they're going to be reaching their adult stages earlier in the springtime and over a longer period of time. So you're not going to get a clear, distinct peak that forms. So the striped bass larvae, they start feeding. They're going to have they're going to have more food available to them. Recruitment will be okay. It's not going to be out of this world. But you have these cold years. Now you have a bloom that forms. So you have your Eurytemora that's producing new nauplii that are hatching. And these nauplii are going to have a lot of food available to them. They're going to be very happy. A lot of them will be surviving. On top of a lot of them surviving, you're going to have more of them being very delayed in their developmental rate. So they're not going to be reaching the adult stage until much later into springtime. They're all going to be reaching that adult stage around the same time. So you're going to have this really squished peak all of a sudden where it can be a whole bunch of these new copepodites showing up because they were all developing together. And then the striped bass will be there, larvae will be there to feed on them. They'll have plenty of food to eat. And you'll have really good recruitment for that year. Oh, just all what the different years look like. So what I want to leave you with, the two big takeaways here. First of all, the main thing is that what's happening in wintertime to some degree is affecting the recruitment of striped bass. Striped bass don't come into Chesapeake Bay until March or April for a given year, but their recruitment to some degree is predetermined based on what the temperature and how much rainfall is occurring during the winter time. It's huge months delay. So there's not much that striped bass can do. Their success is potentially not a lot to do with what striped bass are capable of doing, more what the environment is doing. And then because of the fact that I'm talking about cold temperatures being a very good thing for striped bass recruitment in the upper part of Chesapeake Bay, at least, because temperatures we know are warming within Chesapeake Bay, might not be the best thing for striped bass recruitment, maybe not. But above all, as I mentioned earlier, I'm the only person who's done really put papers out on this Heterocapsa species. A lot more research needs to get done, especially outside of just me. I'm also hoping to do more research and add to that now that I am here at VIMS and continue to work on this effort. But this is something that's clearly been overlooked and is in need of a lot of focus of research. There's a lot going on here, and that needs to be better understood. And that is where I will leave you tonight. So thank you very much for your time. I greatly appreciate your attention.
All right, thank you so much, Dr. Millette. For those of you in person, please raise your hand and a microphone will be brought to you. And if we have any questions online, Allie will post those for you.
Thank you so much. That was absolutely fascinating. To that very last point that you brought up about more research being needed, you had mentioned earlier in the presentation that part of this may be an artifact of us as scientists just not sampling in those winter seasons when weather conditions are really bad. I'm wondering, what do you think we might be able to do to accommodate that? Do you think maybe remote sensing or like satellite imagery? I know that's been used for some blooms. Do you think that might be something we could use in the future to encourage more winter research?
Remote sensing specifically, unfortunately, one of its big drawbacks is clouds getting in the way, and wintertime's got a lot of cloud coverage. But there are definitely plenty of satellite or remote sensing data from wintertime to show the extent of these blooms. And I would definitely promote that aspect, showing just how the full range of these blooms, because a person can go sample somewhere and collect a sample and be like, I have water here with this bloom in it. but then that remote sensing is an excellent way to really just show you the full coverage of it.
Hello. I have a question from Tor from Virginia. Is there any evidence that high freshwater discharge or extreme weather events could move the Eurytemora and feeding stage striped bass larvae down estuary, like into unfavorable environments, or spatially segregate them?
That extensive research has definitely not been done, but all of the data that I have, and the analysis I've done with the data, does not point to there being a point when flow rates become so high that they become detrimental in this case, because we're not necessarily talking about the flow rates that occur when the striped bass and the Eurytemora are trying to match up. This is a month or two before that. But that's definitely part of it. It's much more complicated than I made it out to be tonight.
Well, when there's a bad year for the rockfish, a couple of years later.
Oh, on that aspect, that is definitely, I would say, outside my area of expertise. It's getting into people who work on fisheries and adding that in of then if you have an adult population that's not producing as many eggs, how that's added into the picture is definitely a part that needs to be added into this. I don't have the information on that at the moment though, but that is an extremely good point.
I know you got the rockfish eating the plankton, but are there other fish that need to be mentioned that interact with the rockfish?
That interact with the rockfish. I definitely did not go that far. What I will say is there's definitely other fish larvae that are being hatched around this time, and I chose to focus on the rockfish, but there's other species that could see a similar relationship, and that is also worth exploring.
But we have a question from Matthew. And would it be reasonable to hypothesize based on this research that limits to striped bass harvesting after a warm and dry winter may be prudent to reduce the risk of a population collapse?
So it would, if it was to be based off of that, it would have to be a couple years delayed, because we're talking about just striped bass that are by the end of the year in their juvenile stage, and they're a couple inches long. So they're not, no one's catching them as they're out fishing. They're not in danger of being removed from the population that way. But a couple years later, you could have that year's cohort could be smaller, and there could be some adaptive management that's changing every year based on how successful the recruitment is.
All right. If there are no more questions, can we have another round of applause for Dr. Millette? And thank you all so much for coming out or joining online, and we will see you again next month. Have a great night.